Hello, in today's video, we shall learn in detail about Oedogonium, a green alga. By the end of this video, you'll have a clear understanding about the characteristic features of Oedogonium and its cell structure, cell division, formation of zoospores, development of Antheridia, development of Oogonium, various modes of reproduction in Macrandrous species and Nannandrium species. Let us begin with the characteristic features of Oedogonium. Oedogonium is a genus of freshwater filamentous, free-living green alga. The filaments of Oedogonium are unbranched and are attached to the stem and leaves of the aquatic plants by means of a holdfast in the earlier stages, while in the later stages they may exist as a free floating mass. Some are epiphytic on aquatic plants. Oedogonium terrestris is a terrestrial form and grows in moist soil. The cells of Oedogonium are cylindrical and are characterized by one or more ring-like caps immediately below the cross wall. Every cell of the algal body is similar in shape other than the uppermost apical cell and the lowermost holdfast cell. Each cell of the filament has a three-layered cell wall, innermost made up of cellulose, middle made up of pectose, and outermost made up of chitin. The layers give rigidity and protection to the cell. The thin plasma membrane surrounds the cytoplasm, a nucleus, a central vacuole, reticulate chloroplast, pyrenoids, and other cell organelles. Oedogonium species exists in two forms: Macrandrous type, in which the male and female sex organs, Antheridia and Oogonia respectively, are borne on the filaments and are of the normal size. They may be monoecious or dioecious. Nannandrium species, in which Antheridia and Oogonia are morphologically different. The male cells in this case are dwarf. Cells are vegetative and take part in photosynthesis and growth of the filament. Vegetative reproduction occurs by fragmentation. Asexual reproduction takes place by means of multiflagellate zoospores, Aplanospores, and Akinetes. Sexual reproduction in Oedogonium is advanced oogamous type. Now, let us look into the structure of the plant body or thallus. The plant body is a thalloid, green, multicellular, filamentous, unbranched structure. The cells of a filament are placed one above the other in uniseriate row. The thallus is made up of three types of cells: apical cell, intercalary cell, and basal cell or holdfast cell. The apical cell is the topmost cell, rounded or dome-shaped towards the apex. It is green in color and takes part in photosynthesis. The intercalary cells are all similar and lie between the apical cell and the basal cell. They are longer and cylindrical in outline. Some intercalary cells show the presence of ring-like structure in their upper part called the apical cap. The basal cell or the holdfast is narrow at the base and expands to form a simple flattened disc-like multi-lobed structure. It helps the filament to get attached to the substratum. Next, we shall learn about the cell structure of Oedogonium. The vegetative cells of Oedogonium are cylindrical and surrounded by a thick, rough and rigid cell wall. The cell wall is made up of outer chitinous layer, a middle layer of pectin and an inner layer of cellulose. The thin plasma membrane encloses the protoplasm, which contains cytoplasm, a central vacuole, a single nucleus, reticulate chloroplast with many pyrenoids, mitochondria and other organelles. The large vacuole is filled with cell sap containing excretions, secretions and inorganic compounds. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, we shall try to understand cell division in Oedogonium. Growth in Oedogonium takes place as a result of cell division. It is mainly intercalary but may be terminal at times. The nucleus first shifts from the periphery towards the center of the upper part of the cell. The cytoplasmic material forms a thickening in the form of a ring around the inner wall at the upper end of the cell. Nucleus undergoes mitotic division, forming two nuclei. Microtubules develop between the two nuclei to form the future septum. The ring-like thickening elongates and divides the parent cell towards the apical region. It forms the cuticle of the daughter cell. The distal end of the upper cell contains a small portion of the old parent wall, which forms the apical cap. The floating septum develops into a mature cross wall. The upper daughter cell is known as the cap cell and the lower cell is the sheath cell. Number of caps on a cell indicates the number of times a cell has divided. Next, we shall learn about reproduction in Oedogonium. The different methods of reproduction in Oedogonium are vegetative, asexual and sexual methods. Oedogonium reproduces vegetatively through fragmentation. Fragmentation in Oedogonium may take place either due to accidental breakage of filaments or dying or dehydration of intercalary cells, or due to formation of zoospores and liberation because of wall getting split. Each broken fragment, which are detached from the thallus by cell division and growth, develops into a new filament under favorable conditions. In Oedogonium, asexual reproduction takes place by the formation of zoospores, Aplanospores and Akinetes. In Oedogonium, zoospores are formed singly within the intercalary cap cell called zoosporangium. It contains abundant reserve food material. The contents of the cell recedes from the cell wall and becomes a rounded mass. The nucleus moves towards the periphery and the vacuole disappears. A hyaline region develops, which later forms the anterior region of the zoospore. At the margin of the hyaline region, blepharoplast granules develop and form a complete circular ring. Each granule gives rise to a single flagellum. Several flagella are formed around the base of the beak-like area of protoplast. When the zoospore matures, the sporangium wall ruptures near the apical cap region. A vesicle containing mucilaginous substance surrounds the zoospore. The vesicle later disappears and the zoospore is set free into the water. The zoospore is uninucleate, multiciliate, ovoid, spherical or pear-shaped structure. The anterior end is hyaline and flagella are seen just beneath it. Such an arrangement of flagella is called stephanocont. The zoospore settles down and comes to rest by its hyaline anterior end. The flagella disappear at this stage. The cell divides into two, forming a lower holdfast cell and an upper cell. The latter divides repeatedly by transverse division to give rise to a new filament. Akinetes are swollen, thick-walled, modified vegetative cells formed during unfavorable conditions. They are formed in chains and have reserve food material in the form of starch and oils. During favorable condition, they germinate directly into a new filament. Aplanospores are thin-walled, non-flagellated, non-motile spores formed during unfavorable condition. During favorable condition, they come out of the parent cell and form new filaments. Next, we shall study about the sexual reproduction in Oedogonium. Sexual reproduction in Oedogonium is an advanced oogamous type. The gametes are produced in highly specialized cells called gametangia. The male gametes or anthrozoids are produced in Antheridium or male gametangium, and the female gamete or egg is produced in Oogonium or female gametangium. Two anthrozoids are produced in each Antheridium and only one egg is produced in each Oogonium. There are two types of Oedogonium species, Macrandrous species and Nannandrium species. Macrandrous species, the male is of normal size and the Antheridium develops in the filament of normal size. The plants may be monoecious or dioecious. In monoecious species, example, Oedogonium fragile, Antheridia and Oogonia develop on the same filament. In dioecious species, example, Oedogonium gracileus, the Antheridia and Oogonia develop on different filaments. The Antheridia are intercalary as well as terminal. In fact, any cap cell of the filament can act as the antheridial mother cell. The antheridial mother cell undergoes transverse division, giving rise to an upper, smaller antheridial cell and a lower, larger sister cell. The sister cell divides several times and produces a chain of antheridia. The nucleus of the Antheridium divides into two and the two nuclei lie side by side. Each nucleus is surrounded by the cytoplasm and forms anthrozoid. Two anthrozoids are developed from each antheridium. The anthrozoids are unicellular, uninucleate, and multiflagellate structures. The antheridial wall ruptures and the anthrozoids are liberated. They are surrounded by a vesicle which sooner or later disappears and the anthrozoids are set free. They are similar in structure to zoospores except that they are smaller in size. The oogonium may be intercalary or terminal. Any cap cell of the filament can act as the oogonial mother cell. The oogonial mother cell divides to form an upper cell oogonium proper and a lower cell called the supporting cell. An oogonium is much larger than the ordinary vegetative cell, spherical with a single egg or ovum. Its protoplast moves away from the cell wall and the nucleus comes to lie in the center. A receptive spot is present on one side of the oogonial wall. With the development of the receptive spot, an opening is formed in the oogonial wall and the anthrozoids are attracted towards the egg due to a chemical substance or mucilage. Only one male nucleus or anthrozoid enters the egg from the side of the receptive spot. After plasmogamy and karyogamy, the male and the female nuclei in the egg fuse to form a diploid zygote or oospore. The oospore gets surrounded by 2 or 3 walls. It is spherical and recedes from the wall. The oospore is liberated by the degeneration of the oogonial wall. It undergoes a period of rest. During favorable condition, the diploid oospore undergoes meiosis to form 4 haploid daughter nuclei which are surrounded by the cytoplasm. Each daughter protoplast develops flagella around itself to form the zoospores or meiospores. The oogonial wall ruptures and the zoospores are liberated. They are surrounded by a delicate vesicle which later disappears and the zoospores are set free. The zoospores lose their flagella and come to rest by their anterior end. Each zoospore divides into a lower cell and an upper cell. The lower cell forms the holdfast, while the upper cell, by repeated divisions, gives rise to a new filament. In monoecious species, the meiospore develop into similar filaments. In dioecious species, out of the 4 meiospores, 2 meiospores develop into male filaments and the other 2 meiospores into 2 female Oedogonium filaments. Now we shall study about the Nannandrium species. The male cells are dwarf and are known as dwarf male or Nannandrium. The dwarf males are formed by androspores which are produced in androsporangia. Nannandrium species are of 2 types, Gynandrosporous type and Oedogonium type. In Gynandrosporous type, the androsporangia and oogonia are borne on the same filament, whereas in Oedogonium type, the androsporangia and oogonia are borne on different filaments. Androsporangium. A single androspore is produced within each androsporangium. The nucleus of the androsporangium does not divide. The contents of the androsporangium recede from the cell wall and form an androspore. Androspore is unicellular, uninucleate, motile with a crown of flagella. During liberation, androspore is surrounded by a vesicle which soon disappears and is set free. It attaches itself on the wall of the oogonium as seen in Oedogonium ciliatum, or may rest upon the supporting cell as seen in Oedogonium concatenatum. Next is germination of androspore. The androspore elongates and cuts off a few cells at its apex to form the Antheridia. The lower rhizoid-like elongated cell is called the stalk. The contents of the antheridial cells divide and each cell gives rise to two anthrozoids. The anthrozoids are unicellular, uninucleate, pear-shaped with a crown of flagella at the base of the beak-like anterior end. The anthrozoids are liberated in a similar fashion as found in Macrandrous species. The development of Oogonium is similar to Macrandrous species. Prior to fertilization, anthrozoids are attracted to the mature Oogonium by the secretion of a mucilage. Only one anthrozoid enters the Oogonium through the pore and fertilizes the egg, resulting in the formation of a diploid zygote or oospore. The zygote recedes from the oogonial wall and secretes 2 to 3 layered wall. It is known as the oospore, which undergoes a period of rest. During favorable condition, the oospore divides meiotically to form 4 haploid daughter nuclei which get surrounded by cytoplasm. They develop flagella and are called meiospores. The oospore wall ruptures, liberating the meiospores which are surrounded by a vesicle. Soon the vesicle degenerates and the zoospores are set free in water. After some time, they withdraw their flagella and germinate into new haploid Oedogonium filament. So today, we have learnt in detail about the characteristic features of Oedogonium, its cell structure, cell division, various modes of reproduction in Macrandrous species and Nannandrium species. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.